Welcome, everyone. I am Michelle Krask, and it is my pleasure today to introduce Dr. Diego Pizzagalli, who I've known and worked with for a number of years. Dr. Pizzagalli received his PhD in psychology from the University of Zurich and completed postdoctoral training both in Zurich and at the University of Wisconsin-Madison under the mentorship of Dr. Richard Davidson. He moved to Harvard in 2002, where he is currently Professor of Psychiatry in the Department of Psychiatry at Harvard Medical School, and is also Director of Research in the Division of Depression and Anxiety at McLean Hospital. Dr. Pizzagalli is one of the world's leading researchers in the area of mood disorders, and particularly anhedonia, which he investigates using a multidisciplinary approach, including behavioral, electrophysiological, neuroimaging, positron emission tomography, and molecular genetics. He developed a lab-based measure of reward responsiveness, a key component of anhedonia, that has really become a gold standard in the field. And using this measure, he has made very significant advances in our understanding of the behavioral and neural underpinnings of anhedonia, which I'm sure he'll talk with us about today. Dr. Pizzagalli is a prolific researcher with over 270 peer-reviewed publications and exceptionally substantive and continuous funding from multiple sources for his research. He's editor-in-chief of Cognitive, Affective, and Behavioral Neuroscience, and in recognition of his stature in the field, is the recipient of NIMH Merit Awards in 2016 and 2021, a NARSAD Distinguished Investigator Award, the Joel Elks Research Award, and many others. And he is one of the world's most highly cited researchers, according to the Web of Science. Above all, he is dedicated to methodological rigor and replicability of science, and to collaborative models that enable other researchers to really benefit from his expertise and knowledge. We are very fortunate to have Dr. Pizzagalli present to us today. Thank you, Dr. Pizzagalli.
Well, thank you so much, Dr. Krask, for this very generous introduction. And also, really, I'm grateful to be with you today, at least virtually. The nice slideshow at least shares some of the beautiful campus that you have. And again, it's a pleasure for me to have an opportunity to present to you today. So, as Dr. Krask mentioned, I've been focused on studying the neurobiological underpinnings of depression, but especially anhedonia. And you know, when I moved to McLean in 2010 to start a new center focused on depression, one of my first weeks I actually went to one of our units treating patients with depression. And I was struck by the extraordinary difficulty that you know, those among you treating patients often face, especially for patients with depression who present anhedonia, this profound loss of pleasure, decreased motivation, and really the difficulty that my friends and colleagues, clinicians, had in treating this condition. And of course, I don't have a magic solution. But I hope that our research, contribute, as you will see also toward the second part of my talk, might hopefully contribute to one day, help actually provide some potentially useful information to make a treatment decision for depressed subjects, but particularly those with anhedonia. So let me start by first acknowledging my disclosure. Everything I'll be showing today has been funded by NIH or NARSAD, but these are additional sources of funding that I've received. I have done and I do some consulting for with biotechs and different pharmaceutical companies. Critically, I'll be showing today several funding derived by the probabilistic reward task, which is the methods that Dr. Krask mentioned. And I'd like to emphasize that this task has been licensed to a company called Blackstone Therapeutics, recently relaunched as Neumora. But again, everything I'll be showing you today is derived from our studies funded by NIMH. So this is the roadmap for today's talk. In the first part of my presentation, I'll introduce the concept or construct of reward learning, which I define people's ability to learn from prior rewards and adapt behavior accordingly, which I think we have been studying is actually cross species, as you'll see, and I think it could be a biologically supported phenotype of major depressive disorders. And then I will actually show you how potentially this type of approaches could be used to, again, hopefully make some treatment decision in depression. And I'll share findings from two main studies. These are a very large study. We were fortunate to participate in the EMBARK study and the FASTMAS studies, and I will explain this study as I go along. And again, how actually some of these measures that we've been studying could potentially be useful for making treatment decision in depression. And then I will conclude by emphasizing some of the implication of this work for patient stratification and hopefully one day personalized treatment. So why focus on anhedonia? Well, the clinicians among you in the audience, of course, know this literature or have seen these patients. Basically, anhedonia has been associated with a host of negative outcomes. It predicts depression years later. It predicts poor response to antidepressant treatment, psychotherapy, including cognitive behavioral therapy, even to neurostimulation. So it's been really associated with a host of negative, or poor disease trajectory. And so, again, we were very interested in trying to understand anhedonia, hoping that this information might help us then also devise better intervention. And without going into excruciating details, I'd like to emphasize that also, there are many ways people might become anhedonic. Of course, we in the field generally often use self-report assessment, and we might be asking patients to evaluate how pleasurable it will be, to be in a specific situation. For example, I might be asking a patient, well, imagine that you are on a beach and you see this beautiful sunset, how pleasurable might be this experience? And this is a completely legitimate question, but the point is that there are many ways how somebody might basically endorse not feeling pleasure in such a situation. Maybe somebody has a difficulty remembering positive emotion. Maybe the recollection of this memory is absolutely fine, but the value of the experience is indeed diminished. And that's why I personally believe that it's really important to parse reward processing or hedonic processing in different subcomponents, which of course also has been strongly advocated by the research domain criterion initiative from NIMH, which again identify specific domains that might be impaired in particular subgroup of patients. And the key here is that some of this dysfunction, in fact, might be associated with partially differential or distinct neurobiological abnormality. For example, typically the liking experience, so to speak, the consumatory pleasure has been linked to more opioid receptors and activation in specific brain areas such as the orbitofrontal cortex, whereas, for example, the expectation of reward has been more strongly linked to dopamine functioning and in particular activity in regions such as the ventral striatum, the accumbens, for example, and so on. And so the idea is that, and this is something that we have been embracing in my lab and other labs, is that even among patients with depression, we might find patients that actually have a particular dysfunction, which again might point to abnormality in specific pathways and circuitry in the brain. And today I'll actually focus especially on, well, quite heavily on this concept of reward learning, but also present some findings that speak to reward anticipation and reward consumption. And so when we started this line of work, of course, our focus was on the mesolimbic dopaminergic system, which is shown here, which originate from the ventral tegmental area and project to both the ventral as well as the dorsal striatum, regions such as the nucleus accumbens and the caudate and putamen, and then projects up to the prefrontal cortex. And what we know from really decades of preclinical research and also neuroimaging research is that this system is so important for learning from reward and also for incentive motivation, or if you wish, the wanting component of reward processing. And when we started the design of work, of course, we hypothesized that patients with depression, but especially anhedonia, would show dysfunction in these pathways. And when we started, as Dr. Kras mentioned, we thought that this was about in 2002, when basically I started my first lab in the psychology department at Harvard. I was very interested in develop a method that could actually objectively assess people's ability to learn from reward. And especially something that, of course, goes beyond self-report assessment, something that could also be given to animals, and accordingly could actually increase the translational impact of this work. And what we ended up developing or modifying a prior task, which include the following kind of procedure manipulation. So it's a computerized task, and I'll show you an example in the next slide. But conceptually, what we're showing to participants, we're asking participants to make a decision whether one stimulus or another stimulus has been presented very, very briefly on the screen. And on purpose, these two stimuli are remarkably similar to each other. And again, I'll show you an example in a second. And unbeknownst to participants, what we're doing during the task is that we are rewarding correct identification for one stimulus 3 times more frequently compared to the other stimulus. So there is a very ambiguous stimulus. The subject is not 100% sure what has been presented to them. And we are basically shaping their behavior toward one stimulus over the other by giving this 3-to-1 ratio of reinforcement, in this case, monetary reward. And what we and many other people have shown is that pigeons, well, we have not worked with pigeons, but people have shown this, decades ago. Pigeons, mice, rats, humans, non-human primates, under the circumstances, healthy organisms, healthy humans actually developed a response bias toward the most frequently rewarded stimulus, because there is ambiguity in the signal, and if your brain reward system is so to speak at normative levels, you will develop a response bias toward the more frequently rewarded stimulus. And so we developed this task in humans first. This is actually a representation of what the task looks like. So we present a facial stimulus, a schematic faces, without a mouth first, and then a mouth is presented for 100 milliseconds, so remarkably fast. And as you can see, the difference between the two is remarkably small. And we ask the subjects, hey, did you see the short or the long mouth? And then we present a reward, again, using a 3-to-1 ratio. And in this case, let's say that the short mouth is disproportionately rewarded. Later on, together with the late Athena Marcoux and Andrew Deravakian, we developed a rodent version of this task using auditory tones. Again, we train animals to associate one lever with a short tone, the other lever with a long tone. When they learn the discrimination, we make the tone duration even closer to each other. And here the animal actually, the rodent will receive 3 times more food pellets when they correctly identify one stimulus over the other. And again, if you now use signal detection theory to calculate response bias, if you wish, the preference for the stimulus that has been paired with more reward, which is calculated using this formula, you see that healthy controls developed a very nice response bias. You see the formula here. A high response bias arises when people tend to correctly classify the so-called rich stimulus, which is the one paired with more reward, and they actually misclassify the lean stimulus as being the rich one, right? So again, the behavior has been shaped toward the stimulus paired with more reward. You can see here from these slides, also basically if you look at pure accuracy, you see how people tends to become more and more correct in recognizing the rich stimulus, and they become increasingly incorrect in recognizing the lean stimulus, again, because their behavior has been shaped by the asymmetric reinforcement schedule. And again, using auditory tones, we were very delighted to see that actually different strains of rats show exactly the same behavior, and even psychometrically, and we work hard to make sure that there was actually similar accuracy level basically across species. You see that also in these rats, they become better and better at recognizing the rich stimulus, and actually they become less accurate in recognizing the other stimulus. So again, also rats developed a response bias. I should say that these days, really thanks to a very talented behavioral pharmacologist, Brian Kangas, we have now different versions of this test using touchscreen technology. I'm still amazed just to see these rats using touchscreen technology, also small non-human primates. And as you can see, these days we're using actually lines so that we are working in the visual modality across species. Again, we train animals to basically associate a virtual lever with the long lines and another levers for the short line, and we provide 3-to-1 ratio, and these animals developed a very strong response bias, actually very fast. We can train them much faster compared to the other auditory tones, which is quite remarkable to me. And very briefly, these days we're running often experiment across species using the same pharmacological challenges. And these are some of the findings that we recently published. For example, that ketamine, which of course is a kind of rapid antidepressants effect. We found in non-human primates increased response bias, and other manipulation that we know increase dopamine, such as amphetamine, also very nicely dose dependently increase response bias. And again, these days, we basically are going across species in both directions quite frequently in our studies. So what do we see if we apply these tasks to patients with depression? This was our first study published in 2008 in unmedicated MDD subjects. Healthy controls, again, developed very quickly a response bias, and patients with MDD failed to do so. We have replicated this finding now in different samples, including one in which we found that a low response bias or reduced ability to learn in this task actually predicted chronicity of depression in a naturalistic study of inpatients, a study that was actually conducted in collaboration with Elske Vries and colleagues in Belgium. So suggesting that this type of measure actually could be used to potentially predict response to treatment. What was interesting in follow-up study that we did, and actually for us at least, it was internally consistent, is that this finding was completely driven by the presence of anhedonia and also by the presence of a melancholic subtype of depression. Meaning, and again, this will be very clear to the clinicians in the audience, is that not all patients with depression have anhedonia, And an inability to learn from reward is really driven by those patients that actually have anhedonia, report anhedonia in their life, or actually meet criteria for the melancholic subtypes of depression, which of course is characterized by anhedonia. Also, we have found in other samples that response bias in this task correlates with current anhedonic symptoms and predict future anhedonia. Very briefly, just a couple of some findings that help us kind of build our neurobiological model, if you wish. So again, if this behavior that I just showed you, this ability to learn from reward, is potentially is associated with the functioning of the mesolimbic dopaminergic system, we argued, or we thought, hypothesized that if we use manipulation, pharmacological challenges that, based on the preclinical literature, might decrease dopaminergic signaling, we would expect blunting in response bias. And that's exactly what we saw, both in humans as well as in rats. What we did in across species basically was to use a very low doses of pramipexole, which is a D2, D3 agonist, which is used as an anti-Parkinson drug. It has been used at high doses also as an antidepressant, mostly in open label studies. And but what's very interesting with these compounds is that there is abundant preclinical studies showing that if you use them at very low doses, and we used a 0.5 milligram dose, these compounds actually have a really paradoxical effect, meaning that at these very, very low doses, they are thought to preferentially activate the presynaptic autoreceptor, which is of course inhibitory in nature, and accordingly they have been found to decrease dopaminergic signaling in the mesolimbic system, in the VTA, ventral tegmental area, or in the nucleus accumbens. And in fact, across species, we saw that any rodents or humans receiving, healthy humans receiving a single low dose of pramipexole were very blunted in their ability to learn from reward. Conversely, if we use a manipulation that, based on the preclinical literature, we know should actually potentiate dopaminergic signaling, such as, for example, giving a nicotine patch to non-smokers or giving amphetamine to rats, we actually see a potentiation of response bias. So now we can actually upregulate this behavior by increasing dopaminergic signaling, at least based on the preclinical literature. The finding in amphetamine has been replicated in several labs now using our PRT task. Quite interestingly, now if you study smokers that actually, like heavy smokers, and you ask them to refrain from smoking for 24 hours, you see quite a profound blunting in their ability to learn from reward. Whereas when we test them with nicotine on board, they show normative response bias. These kind of abstinence-driven blunting in response bias was particularly strong in smokers with a history of depression. And similarly, if we administer nicotine chronically to rats and then take it away for 24 hours, we see profound reduction in response bias. Raising the possibility that perhaps nicotine, at least for some smokers, might be a self-medication attempt to alleviate anhedonic symptoms, which I think could be interesting and important to consider in smoking cessation trials. And finally, what we found very interesting is that, again, the ability to learn from reward, which is again shown here by plotting the response bias across blocks. Actually, if we capture the overall amount of learning that people do by just taking simple difference between response bias in block 3 minus block 1, we actually see that this behavior show a pretty Gaussian or normal distribution. Basically, there are individual differences even among healthy controls in people's ability to learn from reward in our task. And what we found recently in a multimodal imaging study is that the ability to learn from reward in our task was actually associated with dopamine transporter binding using a PET tracer called carbon 11 ultrapane, such as those healthy controls with more binding potential for dopamine transporter, i.e., most likely a faster reuptake of dopamine, were actually less able to learn from reward. Or said it differently, healthy controls that actually had a lower binding potential, i.e., actually there was probably more dopamine that that could act in the synaptic cleft, were also the one that learned the strongest in our task. And also in the same study, we found that the functional connectivity during resting states between two nodes of the brain reward system, the bilateral nucleus accumbens shown here, and this region in the ventromedial PFC, such connectivity was actually very nicely, very tightly associated with their ability to learn from reward. That is, subjects that during resting state have more crosstalk, or if you wish, functional coupling between these two key nodes of the brain reward system, were also the one that were better able to learn from reward. So the question that we asked then in a subsequent study was that, well, what are the potential neuromechanisms underlying this inability to learn from reward? And in a series of fMRI study, we use a slightly different paradigm called the monetary incentive delay task. It's a slightly different task, but but what's happening in this task is that people see cues that tell them that there is the possibility of winning money or losing money, and then they actually find out if they won or lost. And what we found in an early study was that during both the anticipation of reward, but not a punishment or a no incentive trial, as well as when they received reward, but not a punishment or a neutral outcome, patients with depression had a decreased activation in this region, in the striatum. So putamen, nucleus accumbens, as well as cortex. So multiple example of blunted responsiveness to reward-related cue in unmedicated individuals with major depressive disorders. But then what we told ourselves is that, well, if we are hypothesizing that perhaps this type of blunting, both the behavioral blunting that we've been seeing and now also in this imaging study, whether this might actually be due to low level of dopamine. Can we actually devise a study to show that if we are putatively increasing dopaminergic signaling, that this type of abnormality would actually disappear, at least momentarily? And so what we decided to do was actually to do the opposite study as the one that in which we use pramipexole, right? Meaning that we decided to basically target the autoreceptor, presynaptic autoreceptor, but now the goal would have been to try to block the autoreceptor, if you wish, so that we are removing the inhibition on the presynaptic cells and supposedly increase dopaminergic signaling. And so the big question is that, if there is such a manipulation that, based on the literature, allow us to increase at least momentarily phasic dopaminergic signaling, will we be able to rescue some of the neurobiological blunting that we kept seeing in our studies? And the solution that we took was to use a single, very low dose of the D2, D3 antagonist amisulpride, because there is some really exquisite preclinical literature showing that at low doses, these compounds have, again, paradoxical effect. They've been found to increase dopaminergic synthesis and dopamine release in rodents, and have been found to have prohedonic effects. And in humans, there was a study by Joachim et al. In Germany showing that low doses of amisulpride potentiated reward-related striatal activation in healthy controls, And some of you might be familiar with this literature. There is actually quite a robust literature, mostly open label studies, showing that low doses of amisulpride, that is 50 milligrams per day, can actually have antidepressants and anti-anhedonic effects. And just to remind you, the amisulpride is an antagonist, is used in psychosis, but the starting doses for in psychosis is actually 400 milligrams. So we wanted to use something that was really far away from the dose found to basically block the postsynaptic receptor. And we decided to use 50 milligrams because, again, there were many clinical studies showing clinical effects using this dose in depressed participants. So what we decided to do, ended up doing, was actually to recruit almost 90 people. Everybody was unmedicated. This was a between subjects design. So about half of the patients were randomized to receive amisulpride, the other half placebo, and the same for the healthy controls. And this is actually a graph of the plasma level of amisulpride. So it actually has two peaks, one at around about 1.5 hours after administration, and then a second one sustained, 3 to 6 hours later. And what we ended up doing was that at this first peak, we basically administered the monetary incentive delay task while people were in the scanner. And outside the scanner at this second peak, we gave a reward learning task. And this is one of these cases where you write a grant and 7 years later, after a lot of recruitment and effort, your hypothesis actually turned out to be what you said in the grant, which is nice. Basically, what we found was that what I'm showing here in blue are the participants receiving placebo. In red, amisulpride. This is the MDD subjects, healthy controls here. And again, as hypothesized, we found that, in fact, patients with MDD receiving amisulpride were completely undistinguishable from healthy controls receiving placebo. And also in the accumbens and putamen, you see how patients receiving amisulpride showed significantly higher activation to reward-relating cues compared to patients receiving placebo. Note how the healthy controls actually behave differently. This drop is not significant, but perhaps an indication that manipulating dopamine in a normative brain, either, well, it didn't have any statistical effect, but if anything else, actually made the performance worse. But this was really the key finding in this particular study, that these low doses of amisulpride, again, supposedly increasing, at least momentarily, dopamine signaling, rescue some of the blunting that we have been seeing in several studies. But this finding, again, is just does not necessarily obviously show that dopaminergic pathways might be necessarily implicated in depression. And accordingly, in the next set of study, we turn to positron emission tomography, again, to more directly measure the dopaminergic system in depression. And there are many ways that one could study the role of dopamine in depression. But what we decided to do in this particular study was to evaluate level of dopamine transporter, with the hypothesis that if indeed dopamine is actually decreased, if you wish, chronically decreased in people with depression, is that we should actually see these as a potentially secondary compensatory downregulation of the dopamine transporter, meaning there are low level of dopamine, and we know from pharmacological study and preclinical study that under these circumstances, one compensatory mechanism happening is that the dopamine transporter becomes downregulated, right? So with the idea that this low level of dopamine, at least let's try to have this level of dopamine act a bit longer in the presynaptic cleft. And there are many preclinical studies which have shown this type of compensatory downregulation. And so when we started the study, we actually had very, very little human evidence that maybe the dopamine transporter might be downregulated in depression. There were a couple of old postmortem studies pointing in that direction, but sometimes actually not even implicating the striatum. There were quite good evidence from stress-related models relevant to depression, all showing actually decreased level of dopamine transporter in the pathways that we were strongly interested. There were some studies showing that rats that have been bred to have increased vulnerability to depression are characterized by reduced dopamine transporter. But the imaging literature was incredibly inconsistent, with no real signal whatsoever. We think that some of these inconsistent human finding really comes from the tracer that were used. Many of the study use a technique called SPECT that is not particularly sensitive. They use tracer that are basically equally sensitive to dopamine transporter and serotonin transporter. And so we felt that using a different tracer might give us a better chance of testing our hypothesis. And we were lucky to collaborate with a group at MGH who actually had developed this tracer for Parkinson research, and is a really exquisite tracer with respect to, again, testing the dopamine transporter, much more selective for the dopamine transporter over the serotonergic transporter, and also other feature that makes it really an excellent tracer to test our hypothesis. And so what we found in this study, we used a region of interest analysis, meaning that we manually traced on structural MRIs the accumbens, recorded the putamen in every subject, extracted binding potential. Basically, we found that people with depression, as hypothesized, had overall reduced binding potential. Some of these values are negative because we remove the variance associated with age, which is actually huge. Age is very strongly associated with dopamine transporter in the direction that is not too favorable. But basically what we found is that across all striated region, but especially putamen, subjects with depression had decreased dopamine transporter binding. We also found the same finding in the ventral tegmental area. This is a very small region, so we want to replicate this. And then we also found this very interesting correlation or association between number of episodes, lifetime episodes, and dopamine transporter, such as those patients with just a single episode, which were not many in our study, were basically indistinguishable from healthy controls. But with increasing number of lifetime episodes, you see this progression with dopamine transporter binding getting lower and lower. We don't really know whether having a low transporter predisposes you to chronicity and repeated episodes, or whether we are seeing a cumulative effect potentially of untreated depression. This was a cross-sectional study. We have no way of teasing apart these alternative hypotheses. But again, I mentioned how the imaging literature was so inconsistent, and we felt the need to basically try to replicate or corroborate this finding using, if you wish, the gold standard, which in this case is actually studying level of dopamine binding directly in postmortem tissue. So these were samples obtained from 15 depressed individuals who sadly all died by suicide, and they were compared to 15 healthy controls that basically died by natural causes. So there is an important confound here. All people with depression died by suicide, and none of the healthy controls died by suicide. So suicide is not equivalent to depression, but that's often the case. This is often the reality of some brain banks. And so we were very fortunate to collaborate with Sabina Beretta and Gustavo Turecki on these postmortem studies. And I know that there's a lot of data here, But the take-home message is, in fact, is that especially for the mature form of the dopamine transporter, but also some other forms that can be found, indeed, MDD subjects had significantly reduced dopamine transporter, specifically in the putamen, which is again the region that we found that showed the PET difference in our study. And also the difference in terms of tyrosine hydroxylase. So basically a convergence of finding between the postmortem and the PET imaging study. So what I've shown you so far is that reward learning is associated with individual differences in frontostriatal and dopamine markers, can be potentiated by pharmacological challenges that we think are increasing dopamine, can be reduced by manipulation that we believe decrease dopamine transmission. We have done also study using chronic social defeat in rats, which I don't have time to show you today. And is reduced in individuals with MDD, but specifically those that have high anhedonia or melancholia. And so based on this finding, we then started really getting interested in treatment studies. And basically the very simple or potentially simplistic hypothesis that we did in several grants and papers was the following. Well, if anhedonia is associated with these reduced functional, structural, and neurochemical markers of dopamine, and if we know that dopamine is so important for many reward-related function, would it be the case that patients that we study that present this anhedonia phenotype actually might preferentially benefit from treatments hypothesized to increase dopamine signaling? So we basically, if you wish, borrowed by the traditional pharmacological model that, well, if you have a dysfunction, try to give something that will ameliorate this dysfunction, right? And in this case, will increase dopamine signaling. And this is exactly what we did in several studies. So in our first study, we thought that potentially patients with MDD who fail to respond to SSRI and actually have this anhedonic behavior before they start SSRI treatment will preferentially benefit from a different type of treatment, which, among other things, may be increasing dopamine signaling, right, using bupropion. And to test this hypothesis, again, we were lucky to be part of the EMBAR study, which was a large biomarker study of depression. Let me walk you through the design. Relatively large sample of patients with MDD, everybody's unmedicated, randomized to sertraline or placebo for 8 weeks. If you were a sertraline responder, you stayed on sertraline. If you were a sertraline non-responders, you were switched to bupropion for the next 8 weeks. Conversely, if you were a placebo responders, you stayed on placebo. If you were a placebo non-responder, you were switched to sertraline. And the simple question that we ask is that, well, how about if we assess reward learning abilities before everybody starts treatment? And does this measure actually will help us differentiate basically who will be responsive to bupropion, especially after failing sertraline? And this is exactly what we tested. And this is a case where, in fact, we found the hypothesized interaction. So a drug by response interaction, but lo and behold, it was 180 degrees opposite than what we were expecting. Specifically, patients who had a higher response bias, a higher ability to learn from reward, they basically failed to respond to sertraline, but ended up responding well to bupropion. So exactly 180-degree opposite than what we were expecting. So a more normative response to reward actually predicted response to bupropion after failing an SSRI. Similarly, when we looked at resting state functional connectivity, we found that there was a region. So we basically looked at correlations or coupling between the bilateral nucleus accumbens and the rest of the brain, which region actually show a drug by response interaction. And it was again this region of the medial PFC and rostral ACC. And also here, the finding was 180-degree opposite than what we were expecting. That is, patients that actually had a more normative crosstalk or functional coupling between the accumbens and this medial PFC region ended up responding better to bupropion after failing 8 weeks of surgery. And I'll actually skip this finding and conclude so that we will have time to for some Q&A. We actually now in several studies found that in fact behavioral or neural markers of dopaminergic functioning can be used to predict response to pharmacological treatment that we know have dopaminergic effects. But the direction in 2 studies that we recently published was exactly the opposite than what we had hypothesized, and we discussed this in the paper. That is, a more normative reward learning system predict better response to bupropion. And in a separate study, we actually found that a more normative reward learning system, as well as a mesolimbic system, as measured by fMRI and PET imaging, predicted better response to pramipexole in MDD. And this is, if you wish, a bit puzzling, and we are scratching our head. It's very interesting that we can find these identify variable that predict response to treatment, but it seems that actually it's not a deficiency model. It's almost like a capitalization models that, at least a normative brain reward system has to be present in order to respond to these dopaminergic treatments. And we were not the first one to find this type of data. There was work by Gabriel Dichter, for example, showing that a more normative brain reward system predicted response to behavioral activation. And these findings made a lot of sense to me because, you could say that you need to have a brain reward system functioning in order to engage with behavioral activation treatment. But we really thought that with pharmacological treatment might be different, and we were wrong. I'll maybe skip this point and just go to my last slides, which is, again, a future study. We are very interested in using these type of approaches to identify, again, subgroup of patients that have particular abnormality, both behaviorally, clinically, and in their brain. And we're very interested in using this type of information, again, to make, hopefully, better treatment decision for our patients to try to accelerate their recovery. In several of our studies, we see that, again, a capitalization rather than a compensatory model of change might be a play here, which again was surprising to us, at least in these studies. And of course, you might ask yourself, we are certainly doing so. What do we do for patients that are characterized by markers associated with non-response? We're currently running some study using ketamine, for example, to see whether we can identify markers that could be helpful to guide treatment selection for these more difficult to treat patients. So I'll end here by really acknowledging many people, the names are shown here, that really have made a huge contribution to these studies, our current funding, and my lab. Thank you so much for your attention.
Thank you so much, Dr. Pizzagalli, for a fascinating talk, and I think really exemplary in terms of both being programmatic and mechanistic, to be able to understand the underpinnings of this very important construct, which has been relatively treatment resistant up until now. And I'm talking, of course, anhedonia, and understanding pathways through which we could actually eventually work on more effective treatments. I think also just with reference to your last point about compensatory versus capitalization models, there's a growing appreciation within the psychological research, treatment research area, that you often find more evidence for capitalization than for compensation. Not immediately intuitive, but that does seem to be apparent. There are a few questions here, and a couple are related to thoughts I also had. One of the first questions, obviously your probabilistic reward learning task and the monetary incentive task, which is so core paradigms and such basic fundamental processes, you rely upon money as the primary or as the main reward mechanism, reward outcome. One of the audience members had asked, how does that compare to primary reinforcers like food, which is usually what is done, of course, in animal studies? Do you want to comment on that?
Yeah, that's an excellent question. We have admittedly use monetary reward or secondary reward much more frequently because in some respect it's also easier to administer, to be completely honest. Of course, you can also parametrically vary them, right? The amount of earnings, for example. But we have done a couple of studies, for example, using, if you wish, social reward, like praise, and we basically see very similar effect. This has been mostly behavioral study, not in the magnet. So generally speaking, we've seen similar reward, similar findings, and it's been really mostly a convenience, right? People, of course, have done a beautiful study administering, liquids or food in the magnet. It's just a bit more complicated. Mm-hmm. And for our behavioral task, of course, we are providing quite a lot of monetary reward. Close to actually 120 instances in some tasks. So it's difficult to deliver mm-hmm. So many primary reward. You could also might use satiation. I mean, we are encouraged that again in our preclinical studies, both with small non-human primates or rodents, mice and rats, we're of course using food pellets or another reward, primary reward, and we see very similar findings. Mm. But that's a very important point.
Yes, I think there was a fairly recent meta-analysis on the monetary incentive task using money versus social reward, and I think the conclusion was it's yielding pretty similar results at a neurobiological level as well.
That's right.
Because you would think naturally that the primary rewards would be different, but perhaps not.
I might add, maybe if I may, and some of this research is still ongoing. Some of the study, as I know, you and also many other people are doing. What is really important, I think, is that, of course, we study these behaviors, in a lab, again, using maybe a monetary reward. How does it relate to real life functioning, right? So in many studies these days, of course, we're giving ecological momentary assessment where we ask them, their interaction, social interaction in real life, so to speak, outside the lab. And so some of this work is now, we're finalizing data collection, so we'll see what we found.
Right, and that was actually my next sort of point, that the tasks that are being used, the PRT and the monetary incentive task, are very basic tasks, and the question is how well does that represent what's happening in real life? But the counter to it is your body of research around the neurobiological pathways and the neurotransmitter pathways really support these tasks as being key mechanisms. So we certainly would not want to veer away from it by any means. There's another question here, more of a clinical orientation. How often do you see isolated cases of anhedonia without the symptoms of depression?
Yes, well, one of our talented faculty in the center, Christian Webb, for his K23, actually implemented a behavioral activation study in conjunction with imaging. And, we had the idea of finding anhedonic subjects, adolescents actually, that actually do not meet criteria for depression. So it's possible you find people, obviously, that don't make full criteria for MDD, but have anhedonia. After 4 years or so, we are getting close to data collection. Complete data collection is difficult. Let's be frank. But they are patients or individuals, who don't meet criteria for MDD. but are really profoundly anhedonic. And we were actually, also many other lessons, of course. And actually, there is another very nice question that I see in the Q&A also with respect to, substance use disorders.
Right.
And that's also something that, we have found extraordinarily interesting. Both individuals with current and prior depression that happen to be smokers, when we test them in our task with nicotine on board, they look like healthy controls. Now, if you are somebody with a current depression or a past history of depression and happen to be a non-smoker, as I shown today, they don't look like healthy controls. They have a very reduced response bias. So again, indicating that, and this is not a call for, picking up nicotine or cigarettes, for ameliorating, of course, your anhedonic behaviors. But it seems to me often that at least some individual with MDD or prior MDD, particularly men, might be gravitating toward nicotine or other substances, as a self-medication attempt, because again, their behavior looks a bit more like healthy controls compared to non-smoker individuals with MDD or prior MDD.
Interesting. Well, one final question before we wrap up. As you know, I've been very interested in some of the overlap between threat circuitries and reward circuitries, threat responding and reward responding. And recently we've been finding that anhedonia is related to deficits in extinction, that there's a sort of a weakened extinction effect. And I'm wondering, are you beginning to explore any of these relationships across appetitive and defensive motivational systems?
Yeah, that's a great question and very interesting finding that you mentioned, Michelle. So yes and no, in the sense that over the years we have done several studies, and we just recently had yet another replication that we have seen that if we test even healthy subjects, but especially those that might carry a genetic liability for depression, if we tested them in our PRT during or before and after stress, and this could be a psychosocial stress or could be a naturalistic stressor, we see that, again, even healthy controls, but especially those with this liability, basically develop what we call stress-induced anhedonia. So, a reduced ability to learn from reward, but particularly, if they carry genetic variants that have been linked to depression. So we have now found this in 4 or 5 studies, actually. So we believe it, meaning that even healthy controls, when they are exposed to an uncontrollable, psychosocial stressor, you may have a reduced ability to learn from reward, which may, we're interested in seeing whether they might actually predict future symptoms. So, we have not directly tested, threat processing using a, field conditioning experiment, for example, but we have seen that the stress is a very potent risk factor for anhedonic behavior.
Interesting. Well, with that, I think we have come to the conclusion of this presentation. I want to thank you again for an really outstanding presentation. Thank you so much.
Thank you so much for the kind invitation and the great questions. Take care, everybody.
Bye-bye.